All right, NEET aspirants, welcome. You know that biotechnology and its applications is a super important chapter for your exam. So let's jump right in and do a quick 5-minute revision to make sure all these key concepts are locked in tight.
So, here's what we're going to cover. We'll look at the promise of biotech, its role in agriculture and medicine, check out transgenic animals, and wrap up with the ethical side of things. Basically, we're hitting all the high-yield points from your NCERT syllabus.
Okay, first things first, let's make sure we're all on the same page. What exactly is biotechnology?
You know, the official definition can sound a bit heavy, but really it just means we're using natural science and living things like organisms, cells, even molecules to create products and services that help us out. Think of it as putting biology on the payroll, getting it to work for human welfare.
And the potential here is just massive. I mean, we're not talking about small stuff. Scientists see biotechnology as a way to tackle some of the world's biggest challenges: fighting hunger, curing diseases, and even cleaning up the environment. The stakes are incredibly high.
Now, at the very heart of modern biotech, we have the GMO, or genetically modified organism. This is where things get really interesting. We're talking about plants, bacteria, fungi, even animals, where we've actually gone into their genetic code and tweaked it, giving them brand new capabilities.
So you might be asking, why go to all this trouble to create GM plants? Well, it boils down to some huge advantages. We can make crops way more resilient, able to handle drought or cold. We can reduce our reliance on harmful pesticides, and we can even pack them with more nutrients. A classic example you have to know for NEET is golden rice, which is engineered to have more vitamin A.
Okay, that's the theory. But how does this play out in the real world? Let's head out to the fields and see how biotechnology is kicking off a whole new green revolution.
So, here's a massive problem for farmers everywhere. How do you protect your crops? Pests can literally wipe out an entire harvest. And while chemical pesticides exist, they can be expensive, not to mention harmful to the environment. There has to be a better way, right?
And this is the biotech solution. Look at the difference. On the left, you see a cotton ball totally wrecked by bullworms. On the right, a healthy one from a GM plant. This is Bt cotton, and it's amazing. It basically defends itself. The pest takes a bite, and that's the end of the story for the pest. So, how does it pull this off?
Okay, this mechanism is a super important concept for your exam, so let's break it down. The plant has these inactive prototoxin crystals. When an insect eats them, the alkaline environment in its gut, that's the key part, activates the toxin. This activated toxin then latches onto the insect's midgut cells and pokes holes in them. The cells swell up, burst, and, well, goodbye insect. It's such a brilliant targeted system.
And remember, this isn't a one-size-fits-all solution. It's incredibly specific. Different cry genes target different pests. Now, for your exams, you absolutely need to know these. Cry IAC and Cry IIAB control cotton bollworms, and Cry IIB takes care of the corn borer. Definitely star those in your notes.
All right, so insects are one thing, but what about pests that aren't insects? Take this little guy, a nematode worm called Melodengina incognita. It infects the roots of tobacco plants and can cause massive damage. For this, we need an even smarter strategy.
And that strategy is called RNA interference, or RNAi. This is genius. The plant is engineered to make a special double-stranded RNA. When the nematode feeds on the roots, it takes in this RNA. Inside the nematode, this dsRNA basically finds a specific vital gene and just switches it off. It silences it. If that gene can't make its protein, the parasite dies. It's like a silent genetic knockout punch. Okay, let's switch gears from the farm to the pharmacy. We're moving into medicine, where biotechnology has been an absolute game changer. And there's no better example than insulin. Think about it. For a long time, the only source of insulin for diabetics was from pigs and cattle. The problem? It wasn't exactly human insulin, so a lot of people had allergic reactions. But now, thanks to rDNA tech, we can use bacteria like E. coli as tiny factories to produce insulin that is identical to what our own bodies make. No more allergic reactions. It's incredible. So, how did they crack this? In our bodies, insulin starts out as proinsulin, which has this extra bit called a C-peptide that gets snipped off later. The big breakthrough by the company Eli Lilly back in 1983 was to make the two main chains, the A and B chains, separately in E. coli.
Then, they figured out how to link them together outside the bacteria to create perfect, mature human insulin.
But making medicines is just one piece of the puzzle. What if we could go deeper and fix the problem at its source? That's the whole idea behind gene therapy. Instead of just treating the symptoms, we try to correct the defective gene itself by delivering a normal, working copy.
And the first time this was ever done clinically is a landmark case you have to know. It was for a little girl with something called ADA deficiency, which wrecked her immune system. What they did was take lymphocytes out of her blood, use a retrovirus to insert a functional ADA gene into them, grow these corrected cells in the lab, and then infuse them back into her. Now, it wasn't a permanent cure, she needed more infusions, but it proved that gene therapy could actually work.
Now, this powerful technology of altering genes isn't limited to tiny microbes or plants. We also apply it to animals, which leads us to what we call transgenic animals.
So, why would we create a transgenic animal? Well, there are a ton of reasons. They're amazing for studying how our bodies normally work. We can create models to understand devastating diseases like cancer or Alzheimer's. They are absolutely critical for testing vaccine safety and new drugs. And we can even use them as living bioreactors to produce valuable proteins.
Just think about that last point, producing biological products. That can lead to some really incredible things. For instance, take this number, 2.4 grams per liter. What could that possibly be?
Well, that was the amount of human alpha-lactalbumin found in the milk of Rosie, the very first transgenic cow. Her milk was engineered to be more nutritionally complete for human babies. Just an amazing example of what's possible, and a classic one to remember for your exams.
But with all this incredible power comes great responsibility, right? So for our last section, we have to talk about the ethical questions that come with all this progress.
And this is a perfect case study in what's called biopiracy. For literally centuries, India has been home to 27 documented varieties of basmati rice. Then in 1997, a US company got a patent on a new basmati rice that was derived from Indian varieties. They basically tried to patent something that was built on generations of traditional knowledge. India fought back, and thankfully, that patent was eventually thrown out.
So, to deal with issues like biopiracy and to make sure GMOs are safe for everyone, you need rules. You need a referee. In India, that organization is the Genetic Engineering Appraisal Committee, or GEAC. They're the ones who make the call on GM research and whether a GMO is safe to be released for public use. And that really brings us to the final big picture question. How do we balance it all? On one hand, we have this immense potential to solve huge problems. On the other, we have a deep responsibility to protect our world and stick to our ethical principles. As we keep pushing the limits of what's possible, we have to keep asking, are we doing this responsibly? It's something to think about. All right, that's our rapid revision. Keep up the hard work, and all the best for your NEET preparation.